Hello, my name is Riley Sansoni, and this presentation will be about how the axolotl genome can help improve regenerative medicine studies. To help build a foundation for my topic, I will first be going over some background information, starting with what an axolotl is and a summary of what was recently discovered about them. I will then move into talking about what regenerative medicine is and its current applications. Then, I will introduce the experiments done on axolotls so far, using the recently discovered information, and how the results can lead to future possible applications in regenerative medicine. Finally, I will end the presentation with some possible complications involved with the applications for the future. So, let's start off with what an axolotl is. Axolotls are amphibious salamanders that are mainly found around Mexico City in Lake Xochimilco and Lake Chalco. They are thought to have inhabited central Mexico for the last 10,000 years and are indigenous to the area. Axolotls are naturally dark-colored, but can shift their skin tone lighter as a form of camouflage. The pink or light-toned axolotls, like the one you see here, are the result of breeding by humans, as they are more pleasing to keep as pets. The main point of interest in axolotls is their ability to regenerate entire appendages and vital structures, such as the central nervous system. Because of this ability, they have been studied widely in labs, and their ability is highly sought after for its possible applications in humans. While axolotls are bred in labs, they are still on the critically endangered list, as a result of drainage projects in Mexico that have reduced the sizes of Lake Xochimilco and Lake Chalco. Now that we know a bit about axolotls, let's find out what was recently discovered about them. In 2018, the axolotl genome was completely mapped out by scientists. The main challenge that was overcome by these scientists was finding a way to map out the massive amount of repeat sequences in the genome fast and accurately. The way they overcame this obstacle was by developing an assembly algorithm called Marvel, which helped them avoid sampling bias and also helped avoid creating gaps or misreading the genome. This is an impressive accomplishment, as not only did they develop a new algorithm, but they managed to map out a genome that is 10 times larger than the human genome. The main discovery that also came out of mapping the genome was the identification of the PAX7 gene. Humans have the PAX3 gene to regulate regeneration, such as healing cuts and broken bones. But this PAX3 gene was completely missing from the axolotl genome and was replaced with the PAX7 gene. Having found out that the PAX7 gene is responsible for regeneration in axolotls, scientists now have a clear target for experimentation regarding how regeneration works in the species and can come closer to finding out how it can apply to humans. So with the discovery out of the way now, let's move back and find out what regenerative medicine is and what its applications look like today. Regenerative medicine is a medical field which focuses on replacing tissues or organs. This is a bit different than the traditional practice of medicine, as the traditional focus is with treating the symptoms caused by disease or injury. The three main concentrations within regenerative medicine are tissue engineering and biomaterials, cellular therapies, and medical devices and artificial organs. Now that we know some general information about the field, let's look in depth at the concentrations and their current applications. First, an example of tissue engineering is the creation of artificial skin and cartilage. In order to create artificial skin and cartilage, doctors use a strategy where scaffolds are implanted into the body with the same shape as the tissue that is desired to be formed. This scaffold then attracts cells to its location, which forms into the tissue. This concentration is still relatively young and has mainly only seen success with soft tissue regeneration. As a second example, for biomaterials, doctors use some commonly known objects such as heart valves, artificial joints, dental implants, and contact lenses, among others, to replace respective deteriorating or missing structures in patients. Biomaterials also have a lot of growth potential, as improvements to these implants create even more effective replacements for patients. As a third example, in the cellular therapy concentration, some of the most common practices are blood transfusions and stem cell transplants. Stem cell transplants are actually the main focus of the concentration, as stem cells can be used to repair or reconstruct damaged or missing tissues. Scientists and clinicians are still trying to find better ways to harvest stem cells and use them more effectively, which poses the main goal as of now. Finally, Artificial organs and medical devices in use today are primarily extracorporeal organs such as the lung substitute and assist and the kidney substitute. Doctors and scientists in this concentration are concerned with supporting or supplementing failing organs until a transplant donor is found, as well as finding other solutions to replacing the organs altogether. Having learned about the current applications of the concentrations and some in-depth information about each of them, let's find out what experiments were done on axolotls that can possibly advance this field. Once scientists found out that the PAX7 gene is responsible for regeneration in axolotls, they were able to conduct experiments to find out how regeneration could be stopped by influencing the gene. In order to influence the PAX7 gene, scientists used TALEN and CRISPR-mediated gene editing tools. The reason why they chose to determine what stopped regeneration was so that they could draw comparisons to the PAX3 and PAX7 gene experiments in other animals. The PAX3 and PAX7 gene in other vertebrates play key roles in muscle, neural tube, and neural crest-derived tissue development. If scientists could find out what made the axolotl PAX7 gene unique from the rest by trying to stop its regeneration and comparing the results to similar PAX3 and PAX7 genes, it could result in new information that could give answers as to why the axolotl has super regeneration. In the following experiments, the axolotl PAX7 gene was compared to the PAX7 and PAX3 gene found in the mice. In the first experiment, scientists mutated the PAX7 gene, which resulted in delayed growth of axolotls, a curved body, and an inability to maintain upright posture when compared to the control. Within the first 20 days of development, the axolotls showed normal muscle mass, but in later stages of development, they showed decreased tail and trunk muscle. These late-stage results were consistent with the mouse PAX7 deletion results. Another interesting fact about the experiment was that the mutated axolotls had no limb muscles. In mice, the PAX3 gene is responsible for limb muscle formation, which means that the axolotl PAX7 gene has similar function to the mice PAX3 gene. In summary, this first experiment showed scientists that when the axolotl PAX7 gene is mutated, it shows similar results to PAX7 deletion in mice, and it has similar function to the PAX3 gene in mice. In the second experiment, scientists chose to delete the PAX7 gene in axolotls, which resulted in a neural tube closure defect. These results are also similar to the results of PAX3 deletion in mice, which also results in neural tube closure defects. Overall, the mutation and deletion of the PAX7 gene in axolotls shows results that are similar both to the PAX3 and PAX7 deletion in mice. The next goal for scientists using this information is to find out how the regulation of the PAX7 gene in axolotls enables them to lose the PAX3 gene, which is essential to regeneration in other vertebrates. Once scientists can pinpoint the exact cause or causes behind this phenomena, they might be able to find ways to use the axolotl PAX7 gene expression to induce regeneration in other beings, such as humans. The most desired application that can come out of understanding the axolotl PAX7 regeneration gene is the use of skin grafts on an amputation site, followed by injections that can mimic the axolotl's regeneration, which would result in the reformation of the lost appendage. To illustrate how the procedure would work, I will first provide a quick summary of how axolotl regeneration works, and then go over how human regeneration can be supplemented with axolotl regeneration techniques. When axolotls experience a trauma, such as the loss of a limb, they go through 3 stages of regeneration. In the first stage, the axolotl experiences wound healing similar to humans. Blood coagulates on the site to close the blood vessels, and a wound epithelium forms. However, after about a week, the similarities end, as the first stage ends with a cone-shaped ectodermal cap forming on the wound epithelium. Then, the second stage begins with the formation of what is called the blastema under the epithelium. The blastema is made up of stem cells which have the ability to de-differentiate into more primitive cells and re-differentiate into new types of cells. These stem cells, in conjunction with signals from nerves and surrounding tissues, provide the means for tissue regrowth. The third and final stage is the regrowth of the lost appendage, using the stem cells in the blastema that are coordinated by the nerves and surrounding tissues. As stated before, the similarities between axolotl regeneration and human regeneration ends when axolotls form a blastema. So, if the procedure to regrow a limb were to be created, it would need to allow humans to grow a blastema with stem cells inside, and also find a way to allow communication between nerves and surrounding tissues to create the correct cells for the appendage that is being regenerated. Such a procedure would radically change the field of regenerative medicine, as each concentration in the field would be able to effectively repair or replace any organ or tissue in the human body. A major example would be the regeneration of the spine in paraplegic cases. People who are fated to never walk again due to unfortunate accidents would be given another chance at walking, thanks to the possible effectiveness of the treatment. However, there are always possible drawbacks and complications that can occur with any procedure. So let's look at some of the possible complications that could occur with this one. The two most dangerous complications that can occur with the procedure are cancer growth and tumor formation. Since the procedure deals with cell growth, it is possible that the growth can become uncontrolled and cause cancer. Similarly, tumor formation is also a worry, as if the cells do not differentiate correctly, they may form large tumorous masses instead of the desired appendage, organ, or tissue. Looking forward to the future, scientists will have to be cautious if they want to create the regeneration procedure, as these possible complications can be life-threatening. Similarly, patients might also have to weigh the risks of the procedure if the complications are only able to be minimized. However, there is still much to be learned from axolotls, and the reason for their super regeneration is still yet to be uncovered. Thank you for listening to my presentation.